It is a pleasure to be invited to speak with everyone. I'll take you for a little ride into the engine room of the biochemical assay core of ASAP. Maybe talk about one other project as well. I hope that I might be even a little bit under time, but we'll hope that it fits well. So as John mentioned, I'm working here in the Weitzman for about 12 years. I work at a place called the INCPM. We call it the GINCPM for short. It's got a longer name, of course, which means the Israel National Center for Personalized Medicine. So there's this nice building over here on the left, very picturesque at the edge of the campus. We're kind of not in the center of the Weitzman. It's a very beautiful campus in general, but we're kind of in this little corner. As John said, I'm in charge of basically two units within the INCPM. The INCPM is sort of a technology hub for not just the Weitzman, but for all sort of burgeoning Israeli biotechs and hospitals and academic institutions. So I guess I'd have to define myself mostly as a screener, as a HTS developer screener, but I also do run the medicinal chemistry unit as well, which I hope it'll come out in the talk. What advantage there really is to having these two types of activities sort of married together in a harmonious workflow, which is, I would say, in the academic environment, sometimes is not always a given. So what we have in the INCPM is about 300,000 compounds in our screening library. All the instrumentation and infrastructure do compound logistics for such a large academic library. We do development of assays. We have lots of automation, and we also perform orthogonal and confirmation assays. On the other hand, the medicinal chemistry group is doing reagents for screening. They're producing reagents, or they're also helping with chemical triage and resynthesis of screening hits, and also helping with SAR. So that's it, from a high level perspective, and more in the focus of ASAP. So I guess people are probably familiar with sort of this workflow which we have in ASAP, which is really kind of progressive, different, almost like a biotech pipeline type of environment. And here I've outlined where we fit in with this ecosystem here near London, and I are sort of leading the biochemical core, and we are mostly giving services to Project 2, which is the target enablement wing of ASAP, and Project 3 and Project 4 fragment to lead in target validation and covalent targeting strategies as well. Actually, I should have expanded that circle out to include Project 5, which lead optimization, with oversight on my part, but you can imagine, so it's basically this whole central part of the slide is really where we're engaging constantly. So here's just when I was getting prepared for the lecture, I realized that I didn't know how many clinically approved antivirals there were, and this is not up to date because this is a slide from before the pandemic. But it was encouraging to see that the majority of the antivirals are small molecules, and the proteases are the targets which we're targeting largely in ASAP. These are good enzymes for screening compounds, and they're also, of course, very good targets for antivirals. So the model system, or the focus of our work in ASAP, really resulting from the coronavirus pandemic, has been on the main protease of the coronaviruses. We started, of course, with SARS-CoV-2. We've expanded over to MERS coronavirus as well. But in one project, which I'll show you at the end, we're actually working with other people in the Weitzman also on inhibitors of viral fusion. So that could be a bit of interest as well to this audience. to complete the life cycle. I'm sure everybody knows this, the completing the life cycle of the coronavirus. So you have this proteolytic event of processing the polyprotein, protein, which is essential and has been targeted by nirmatrelvir and other inhibitors, as well as the ones that we're working on. So way back to the beginning of the pandemic, the initial solving of the crystal structure early on, showing us that where the main protease, how it's working, and in terms of its structure. An early inhibitor they called N3, which is a covalent, rather non-specific inhibitor of the main protease. But in this particular publication, which sort of kicked off the whole my whole entry into this world. As a screener, I noticed that they were also publishing together with the structure in HTS of about 10,000 compounds, in which they focused on compounds called like epsilon and disulfiram as hits. And I don't know if Neer's joined us, but we can remember that, I said, well, what is this? Why epsilon and disulfiram? These are, we sort of, as screeners, we consider these to be these are very promiscuous compounds that we tend to try to disregard early on in the process. So I was really kind of wondering about that. And so I think this is a good point to kind of refocus people onto the life of a screener, in which you're talking about the sort of funnel-like approach in which you have lots and lots of compounds. We're primarily focused on having a good screening assay. The compounds themselves, you're trying to collect as many compounds as possible in order to get as many shots on goal, but not necessarily with an ongoing hypothesis. And then the value of the compounds is built as you go further and further through the screening funnel by doing either secondary or counter assays, which we're going to sort of eliminate the less interesting or the more non-specific compounds, the false positives, and then building in orthogonal assays to sort of confirm that the compound is working in the way that you expect it to work and not be some artifact of, let's say, of a screening assay, and ultimately generating something which you can expand around in structure-activity relationships and confirm your hypothesis in a proper medicinal chemistry type of format. So the value of the compounds increases as you go down this funnel, and this is very much the experience that I have day in, day out working with people that are coming to do high-throughput screening. So I kind of went back and in advance of this talk and said, well, what happened to all the HTS that was done around SARS-CoV-2 and MPRO? And I'm not sure this is a completely exhaustive literature search, but I did find that there were quite a few publications, including 1,800,000 compound screen. Here you can see down at the bottom, that was published just a year and a half ago. And we actually did in the Weitzman, we did a 185,000 compound screen as well in the early part of the Moonshot initiative, which we never published. We got, of course, very, very quickly focused on the output of the Moonshot, which was a much smaller compound set. And of course, Ed has talked about this in previous open science forums, so you can go the evolution of the compounds there. But this is just sort of an idea, or sort of to bring home that, on the one hand, yes, drug discovery sometimes is a numbers game. You want to go for the big numbers, but in the way we're applying it in Moonshot which is clearly beneficial, because we have gotten to some very good output, including this recently disclosed DNDi 6510, which is our clinical candidate now. So the way this kind of impacts the screening funnel is that we have in the ASAP and the Moonshot format, we have a smaller screen, which is done in P2, the target enablement structural biology fragment screening part of the process. And then there's going to be a generator, machine generator, not necessarily machine, could be people that are designing compounds, but in the P3 or P4 or P5. And then we're going to be delivered to the biochemical corp actually quite a larger set of compounds than would have come out from the fragment screen. So there is this expansion of potential hit matter which is coming in, but it's not nearly as large as in a typical high throughput screening campaign. Now, what this means for us, in my experience, is that we're getting better compounds than we would normally have in our HTS deck. The compounds are being, driven by the hypothesis, which is being generated early on in ASAP. There's many more eyes that are on the data than I would typically see, and this is, of course, a very educational and exciting experience to hear everybody pitching in and seeing the compounds improve on a weekly basis and engaging with everybody. Further down the funnel, instead of doing these confirmation assays, the compounds are being cycled back to the structural biology core and being crystallized or soaked into the crystals, and then ultimately to the antiviral core as well, where we're seeing actual antiviral activity. So it's reminiscent of the screening funnel that I knew so well, but I think there's these other levels that really drive the value of the compounds. So to remind everybody just that we know that there's the MPRO is our sort of main focus at this point. It's a proteolytic enzyme with this catalytic dyad here, and the way this is translated into practice in the screening assay is that you're going to be feeding it a peptide usually, which is derived from one of the regions of the polyprotein, usually NSP4-5 site, and then it's going to have either a fluorogenic type of fluorophore, which when it's cleaved, it will then give you a fluorescent signal, or these sort of dark fret quenchers in which you actually have the proximity between the two fluorophores is what keeping the fluorescent signal down, and the catalytic activity releases the quenching. There's quite a lot of MPRO substrates around that people have used in these different publications. We don't have to go into it, but they're all more or less the same idea, and maybe they have different traits. We have been using this one down here, which is a fluorescein and a Dabsil quenched peptide. It's I think when we first started in ASAP in MPRO, COVID-19 moonshot, I should say. So NIR's group brought it to us, and that's what we've been using ever since. Now, the reason why as a screener I like this is that it's basically generating a fluorescein-based green fluorescence, which is something which we're very aware of in screening, that you want to use a very bright and somewhat shifted towards the red fluorophore. Many of these fluorophores, like the Sanacoumarins and things like that, they actually tend to have some interference. So we try to get away from these things. So it's worked very well for us, and this is what our day-to-day life or week-to-week life looks like in MPRO. We have this assay running here, and it's already been rather well tweaked. We're getting weekly shipments of compounds from Enamine, which are being designed by the various chemistry teams. We're registering them. We're preparing assay-ready plates using our Echo acoustic dispense system, and then we're going to perform, just a very easy liquid handling process in reading your plates. So very, very straightforward and very amenable to this type of work. This is what a typical screening run from a couple of weeks ago. This is kind of what the data would look like. We're doing multiple plates per run. We're taking the compounds that we're getting, we're plating them out in a dose response. You can see here's one compound which has been placed on different plates in the plate, and this sort of helps us to mitigate some of the trending effects that might have due to liquid handling errors. You can see that there's a high and a low control on each plate. It's giving us a Z prime, which is what the screeners always like to go for to sort of evaluate how their assay is performing. So this is a very good signal. The 0.96 is quite robust assay. And you can see how for an example, how this particular compound translates into the dose response. Very, very tight, almost no error bars between the samples. And this is what we're generating week after week after week on multiple compounds. So if we kind of take a step back and say, well, so how's this performing? What are we doing as we go on with this thing? You can see we're testing typically between 20 and 30 compounds every time they come in, every week or so, coming in from synthesis and enamine. Occasionally we'll get more compounds, depending on whether there's a big library coming through or some sort of initiative which is exploring something in a larger set, but that's no problem for us. And over here you can see how the progress of the project really is going with the average IC50. We're driving down. Obviously, this is not me. This is Ed Griffin's group, mostly, that are turning out wonderful inhibitors that, as we get better and better in our process, the compounds are getting more and more potent. So that's sort of the view of what's going on in MPRO. We typically run on every MPRO assay. We're going to be running this compound, which we affectionately call the Carlson compound, because it was published by Jens Carlson in Uppsala. It's a very good inhibitor, both of SARS-CoV-2 MPRO, but also of the MERS MPRO as well. And down below it, you can see one of the more recently disclosed compounds from Ed's lead optimization group, in which the spirolactam has been sort of merged in here in the center part of the compound. And this is sort of reflects the way our compounds have developed over the course of time. Now we are doing screening not just against SARS-CoV-2, but also against MERS main protease. And this is where the surprise really kind of came, because we thought everything was going to work. We were just going to swap the enzyme out, do some assay development, and it was going to form as usual. And what happened was we immediately got these dose response curves, which had a very big charge or activation in signal, and then inhibition later on as the concentrations went higher and higher. So it took us quite a while before we actually believed that this wasn't an artifact in our liquid handling or process or something like that. And you can see, you can refer back to David Min's Open Science talk from a few weeks ago, where he really went into the details of how you could derive predictive data from even these crazy kinds of curves. We don't necessarily look at this in this way. We're sort of asking the question, how can we harness this phenomenon, which is interesting scientifically, but we ultimately want to be able to peg the compounds and say whether they're more potent or less potent, and then give the antiviral core the best compounds for doing their antiviral assays. So what you can see here is that in two compounds, which the structures have not been disclosed, so I can't show them, but you can see how as the compounds increase in their potency, both in the SARS-CoV-2 MPRO and in MERS, you'll see that although you're getting this sort of classic left shift of the compound of the IC50 dose response in SARS-CoV-2, here you can see actually the peak of activation is starting to kind of crash down towards the line of background. And I can tell you that as the compounds get better and better and better, this activation starts disappearing from our horizon. So it's interesting, but ultimately we're assuming that the activation is essentially a compound-induced transitioning from an inactive monomer to the active monomer of MERS MPRO, and we've there's a good publication which kind of talks about this. So just to kind of see to zoom back out what's going on. So we're getting from target enablement in project 2, we're getting assays and reagents usually. We have a bunch of targets that are sort of ongoing in our core, and we're delivering data to these different projects. So it's usually screening data, potency, IC50. Occasionally we're doing selectivity or biophysical or orthogonal conformations. Also, we're helping the covalent core as well, either plating out compounds. We're actually doing a project as well, doing screening, sometimes with intact protein LCMS. And of course, the very strongly integrated with the lead optimization group. So we're priority is turning working assays into screenable assays, and with a priority in reproducibility and delivering the data and sometimes compounds to project clients. These are the current roster board of the assays and targets that we're running. You can see they're quite high focused, of course, on the fluorogenic M-Pro proteases, but there's plenty of others which are running. We don't have time to kind of go into these things, but things are exciting and there's a lot of activity in the core. So with that, I'm gonna get off of ASAP, and with the last couple of minutes, show you something which is not ASAP at all, but is antiviral related. This is a project which came to us, again, not quite when ASAP, when COVID-19 moonshot started, a little bit later on, but it's coming from Dr. Ori Avinoam, who's a PI here at the Weitzman, and his student, then student Suman Khan. Now, Ori is interested in membrane fusion as a biological process. He said, well, you've got the spike protein, spike glycoprotein. It has to go through these this processing as the SARS-CoV-2 wants to engage with the ACE2 receptor. It has to be processed by the TMPRSS2 protease, and then you have this fusion event. So he was interested in targeting this with small molecules, but of course there's the issue of screening this type of stuff in a phenotypic assay, not with a, you don't want to work with a hazardous virus. I know the antiviral core does this, so we're of course not as brave as those guys. So Noga Kotzer is our high content imager screener in our group, and Krista Schursch is our senior medicinal chemist. So they were very deeply involved in this project. And what you can see here is the pseudovirus screening assay, which Suman developed, in which you're taking cells that are sort of expressing the spike protein and infecting them with a VSVG virus, which is obviously not pathogenic to humans. It's got the glycoprotein, which has been deleted. And as the virus is exiting the initial packaging cell, I guess you would call it, it's taking with it a GFP reporter and the spike protein from the SARS-CoV-2 in place of the VSG. So you're getting basically an infection, which is monitored by a GFP reporter. And then we're going to be doing image analysis to quantify how much infection event has happened. We turned this into a high throughput screen. As you can see, it's in a 384 format. There's a lot of making viruses and transferring viruses and so forth. I wouldn't say it's the pinnacle of high throughput screening, but it certainly enabled us to, over the course of about a year, do a 200,000 compound screen. You can see sort of what the data coming out of the automated microscopy looks like. We have sort of all these infections, a virus which, or without virus, gives you a blank, and then an inhibitor would wipe out or give reduced infection events as well. The nice thing about this this type of screen is that they could swap in and out the different spike proteins. So they designed a triage in which was completely based around the different pseudoviruses. Here, the primary screen, which is done about 200,000 compounds, was done using the coronavirus spike, but then they would counter screen against the glycoprotein from VSVG, which was reintroduced back into the virus, and getting rid of all the things that inhibit both the VSVG spike protein and left with only spike-specific entry inhibitors, and then swapping out for a MERS-derived spike protein. And ultimately, after this whole process, they ended up with 5 compounds of interest, which is quite, from a screening perspective, that kind of gives you the idea that if we hadn't done 200,000 compounds, we probably wouldn't have gotten even, anything like this type of output. Now, what we got early on, for instance, was this compound here called methomostat, which is an inhibitor of the TMPRSS2 protease. So that was both a good, maybe not a beneficial hit for us in terms of, antivirals, even though I think it actually put into clinical trials at some point, but it was confirming the principle of how the assay is working. And then we got these other inhibitors, which were more compounds from the screening deck, and it required quite a lot of I would say medicinal chemistry diligence to make sure that we weren't led astray by some of these compounds. Now, the particular one which I'd like you to focus on here is this 163-855, which, after we kind of looked at it much closer with resynthesis and so forth, we realized that the structure that was, delivered from the screening deck was actually erroneous, and that actually there was a monosulfoxide here in the center. But so we resynthesized this compound now in its proper form, and which came as a racemate, and we got these quite nice, even close to nanomolar inhibition of the wild type, or not the wild type, I think it's the Wuhan, the Omicron spike version. We could as the project went on, we kept on changing the spike for whatever version of spike was the current pathogen of interest. But these compounds were then sent to the Pasteur Institute as well, where they did live virus testing, and they also gave similar inhibition on in live virus. So the pseudovirus and the live virus was reading out quite well, but because this is a racemate, so we went out and further purified to enantiomer pure mixtures, pure compounds, and we could see that this one enantiomer actually gave even higher potency, which is obviously what you would like to see as you get more validated medicinal chemistry type of feedback. And this compound, this enantiomer was essentially devoid of activity. So that's really where the publication kind of ended up. I wouldn't say completely that we're not targeting some sort of host target here. I think that's still, we still left to be proved. But in the meantime, this project is ongoing. We're going to try to make some more compounds around it. So in summary, we're leveraging HTS infrastructure for iterative screening, with the Moonshot and the ASAP consortia. It helps us do rapid turnaround and dissemination of data. We're running multiple targets in parallel, and I showed you an example of phenotypic screening for fusion inhibitors to translate from pseudovirus to actual live virus. And I stress the importance of medicinal chemistry linked with the screening platform in order to mitigate risk. So with that, I think I'm pretty much on time. I'd like to thank everybody who's been helping us, particularly Neer's group, Ori's, lab mates, and Suman, and of course everybody from ASAP who's been producing compounds or reagents, and helping us with the assays that we integrate into the biochemical core. Thanks.